Oh, thanks so much for this nice introduction. Really, thanks a lot. Can you guys hear me? Like, is it working fine? Perfect. So I'm right now stationed in Hamburg at the Center for Natural History at the University of Hamburg. But most of the things I'm going to talk about today have actually taken place, if I can make it work now, sorry, uh, during my time in Copenhagen in the lab of Katrine Worse at the University of Copenhagen. And they were funded by the Velux Foundation and the Danish Research Council. So this is part of the work that we started during my PhD, but then continued on during the postdocs, and which we are kind of dragging into this like new split activity, more or less, between Copenhagen and Hamburg now. So just to give you an idea about the nervous system, I'm not sure how it looks with nervous system knowledge. So this is something that has been really interesting me for a long time. So this is kind of clear, but many people have like this issue with like, okay, worms, do they really have a brain? What? They have nerves? So yes, they do actually. And on the left side, I have like this kind of teaching book picture that like all the students get like at the very beginning. So how does a brain of an annelid look like? And you can see here, there's like the mouth opening here, and around the pharynx, you have like the brain, which is on the dorsal part of the animal, and then the ventral nerve cord. And this is more or less this one stable thing that is found in most of the annelids. But this, of course, is not the entire story, because if you look closer at the annelids, and this is like also talking about the macroscopic ones, this brain looks a lot more complex than people actually think. So what you see on the left side is actually drawings from a paper in 2005, which was summarizing a lot of nervous system architecture. And there are some tracts or some highways in the brain which allow the animals to kind of switch faster or like communicate the information much faster between the two body sides. And here's where the differences can actually happen. So these would be like the A and the D would be like two different species that have been investigated, which showed like some differences in like, for example, how the eyes or how the pulps are working in there. So the most variable parts in the nervous system are like these commissures in the brain, but also like on the ventral nerve cord, when we think of it looking like a letter, then it actually is like the connections between these two sides of the letter that can vary between the species. When we look to meiofaunal annelids now, the thing is that they were described as being much simpler, which most of them also like are, and that they are less complex and possibly resemble an ancestral state like a bit more closely. And this is kind of represented by the drawings here in the lower part, where people try to use like the nervous system architecture to reconstruct phylogenetic relationships within the groups, also like spanning from annelids to crustaceans. What we didn't know at that time was that there's just like a lot of variability going in there. And there is a lot that is kind of hidden by the state of the adult animals. And this is what we basically wanted to investigate to begin with. Meiofaunal annelids, that looks like one more or less concise group, but the problem with annelids is that all these meiofaunal lineages, and there are quite a lot of them, they have emerged independently. So they are nested between macroscopic groups. So you see it here on this like tree in this like violet purple section in the very bottom. Like these long branches, for example, these are the super weirdos, but like them, many of the meiofaunal annelids are related to macroscopic annelids. So what we think now is that like this miniaturization happened several times during evolution. So that also means that each of these like lineages has maybe like found their own way on how to get like small, like how to reduce complexity, but also how to reduce, for example, cell size or something like this. Meiofaunal annelids are also very diverse, which is partly because they have like this different background or this different relationship, but also because they have adapted specifically to their environment. I mean, many of you will know that meiofaunal annelids are very diverse, and they range from like this super, like very small blotch-like animals to the ones with parapodia that can really crawl like with their parapodia between the sand grains or between like bigger structures. Also, they range in size quite a lot. So the smallest annelid that we know now is like the dwarf male of actually this very first animal, which is also something that I'm working with, which is 50 micrometer in length. And then like there are some really big ones that actually are like this, okay, is it really meiofauna because it can wiggle through a sieve or something like this. And now I want to introduce this, like the family that I've been, or like, first of all, I want to go like into this nervous system structure again, saying that within like this meiofaunal annelids, there is not only like this one way to reduce the size, but nervous systems in this meiofaunal annelids can look very different. So on this left side, there's some, like one species which has it even in the name, Lobato that the brain is very lobate. So it looks more or less like a big worm's brain with having these separate lobes and this really concise nerve tracts, which is shown here like in the color coding kind of, but which we can also see by alpha tubulin labeling and confocal laser scanning microscopy. So that means we use antibodies to label cell skeleton structures and can therefore see like these condensed nerve fiber rings, which are like these commissures in the brain. So this looks like quite actually advanced compared to like the one that we can see on the right side, where we need quite a lot of magnification to, and also like a bit of good willing, of course, to look at it and say like, well, there's also like these condensed fiber pathways that we then can construct to like this much simpler layout of the entire brain. So the thing is just like, how much of this can we also really prove, or how much of that is kind of combined to a function that these special tracts actually have. So, and the later one is part of like my very favorite research animals. So it's the one here on the left bottom side, it's Dimorpholus. And in the family of this worm, there are like 3 different kind of morphotypes, and they differ in like the coloring, as you can see here on the pictures on the left side, also a bit like on the size and on the body shape, but most importantly in their life cycle. So like these orange ones, for example, have a super long life cycle of over a year, which has like this very long encystment phase, which is shown here like in yellow. So that means these animals encapsulate themselves, kind of dissolve almost, and then afterwards hatch again as new adult animals. The second one in the list, Trilobodrillus, they look like a bit more slender, a bit more easy. The life cycle is a bit faster. They don't have encystment, and otherwise resemble like the last one quite a lot. But the last one has this super rapid life cycle. So it takes about 1 month, like 3 weeks to 1 month, until they can start reproducing again. And they also have dwarf males. So this is more or less the 3 representatives in this group that we have been dealing with. And nicely enough, when we tried to check for that, sorry. Yeah, we also found out that they actually group in like 3 different genera. So we now can say we have 3 genera which are related to each other, but we can use them, or like use the findings in them to make like evolutionary reconstructions of nervous system development. And like the last one, we can also even hold in culture. We tried with the other ones, but it's a bit more problematic because especially the orange one is, yeah, not taking it too nicely. And the one from this one, the Trilobodrillus, we get mainly from Sylt, for example, and they kind of stay alive for quite some time, but they don't really get into the mood of reproducing. So we're still working on that, but the last one we really can keep in culture and get like all stages of the development in there. So why does this now relate like to nervous system anatomy? We want to know like how does actually a nervous system structure itself, or how does this develop during development or evolution? So when we look at the brain of like this Dimorpholus or Dimorpholus females, they look very unpatterned. So our first thought was like, well, maybe the adult animal has lost all the substructure like lobes, and we could find it when we look like a bit closer. So the first thing we did was actually using Darby staining to maybe find something like mushroom bodies. So like places in the brain where the cell nuclei are very condensed closely together, and which kind of suggests that this part of the brain has a separate function to the other one. Well, we didn't really find that. I mean, what you can see here is like the brain of the worm stained with Darby, which stains the cell nuclei. These holes here is where the eyes are embedded. But the rest of the brain is very homogeneously separated in like how dense the nuclei are, or how packed they are. Next, we tried, when we use like antibodies against different neurotransmitters, maybe we can then find like a pattern that, for example, like certain neuropeptides, which are like the signaling molecules in the brain, are just found in the anterior part or just in the posterior part. Well, unfortunately for us, this also didn't really work. So what you see here is like a model of this brain derived from the Darby stainings, and each of these little of the colors of these tiny bubbles is like a separate neuropeptide, which I mapped like onto this like one brain. And there is actually, the cool thing is there is no overlap between them. So it seems that each cell is very specific for each of the neuropeptides we used. But on the other hand, it also doesn't really show any kind of pattern that we can say like, okay, in the anterior part, there's something else going on than in the posterior part. So we couldn't really assign any functionality to that so far. As a next step, we try to say, well, if we go like one step further down and not just use like this biochemical pattern, but actually go to the molecular pattern and look for genes. Yeah, well, we can see like that there is a differentiation between like the anterior and the posterior parts. So just in the first row of like this picture, you see like a graphical representation of where we found gene expression in like the worm, so like in dorsal, lateral, and ventral view. But the problem is like when we compare it like to what is known from other annelids, we first of all have the problem that it's different stages that are compared or different sizes. And the second problem is kind of, well, we just have one stage. We actually need to look at like younger stages or older stages to just see whether there's any differentiation. Also, the pattern that we see, if it resembles like the larvae, then it is like a very general neurogenic patterning. So it's not something that we can really say relates to, for example, vision or smelling or something like this. But it didn't get us down. So we now actually went back into development and said like, well, if we go like to the critical phases during this development in 2 species, and we chose like the big orange ones and the ones with the fast life cycle, maybe we can find a point where this brain actually looks a bit different than before. And so the problem was like, we didn't really find any regionalization. But when you look like at the big drawings here, then you can see in the ventral nerve cord that the distribution of the commissures, like this transverse connection, is quite different between the 2 animals. So we have in the Dimorpholus, like the white ones, we have like 3 different commissures per segment, while in the orange ones, there's just 1 commissure. And this was always like very kind of, surprising, because why is the one so messy and the other one is all cleared up? And one idea was that this is related to this fast development, because when they have less time to develop, they maybe just have to stop sorting out their own nervous system. And we really found like this upper stage, like this early juvenile, to be like the first one where we can see this difference developing. And like this pre-hatching animals here, these are the ones where we can more or less see a similar pattern between both of them. So that led us more or less to this conclusion that the white ones, most likely, also because they have dwarf males, evolved by progenesis. So that means they have more or less the same development as the orange ones, but at some point they just like arrest the somatic development, so they don't go on with sorting out their systems, but then mature. So that means they actually are like reproductively active animals in like a juvenile body. And with this is like during evolution, this could kind of lead to this body's form. On the other hand, it could of course also be that the orange one just does like overdevelopment, so then actually continues to sort out things. That is something that we can't really prove or disprove right now, but which would really help would be like taking the third species in the mix and also doing this developmental phases and then checking what we can actually find out. That this is something we really want to look into a bit more. So we have this actually really cool system also for evo-devo studies, where we have 3 different species in 3 different genera, which we can use with show like different life cycles, which also are like different from each other, like really nicely set apart. And where we have different brain morphology. And this is like this neuropeptide patterning that I showed at the beginning, now done with all 3 species, where we don't really see anything that's conclusively like the same in all of them, but it's also not really the different, because they look just like, in general, much more different from each other. And then we have like a third system that we can compare, and this is the male copulatory organs. So all these animals have like quite prominent copulatory organs, which they use to penetrate the epidermis of the female and do hypodermal injection of sperm. And at first we thought, when we just looked at the dwarf males, that this is something that is very derived, just because they are like dwarf males and that's an adaptation. But because it takes up more or less like a third of the entire body. But then we looked at the other 2 species, and like the copulatory organ in like all its structure and also in the nervous innervation is very similar between them. So what you can see in this schematic drawings in green is like the muscular, or like one of the muscular layers. So they all have this inner penis cone, which works like a pistol. And then they have the penis sheath around it. And then in gray here is alpha tubulin staining again. So this is like the major nerve tracts that are running around it. And in all species, we have like this one nerve ring, which is actually going around the copulatory organ, which most likely orchestrates this contraction of the musculature. And this is something we wanted to look into a bit more, but then of course the big animals are really big. So we said, why not go like for the small ones? They are of course very small. That means like very hard to work with all the techniques we applied. But on the other hand, there is a really good background on that. So in 1988, Windhoff and Westheide actually did this beautiful electron microscopy study where they sectioned through an entire animal and then reconstructed nerve by nerve and could therefore give like some sort of an idea of the overall architecture. And we're now building onto that, and this is still like ongoing work because we now have the section series, but we're still like into the tracing and mapping all of the synapses. So because we figured out when we trace the neurons, this is one part, so we really know how the nerves look like and how they're extending, but we don't really know where they're talking to each other. So we really need to have the synapses in there as well. And each nerve can have several synapses, or the synapses can be so small that we can't see them. So this is more or less the critical part there. Another thing we're doing also with the dwarf males is doing this immunohistochemical labeling. So using the antibodies against neuropeptides or different neurotransmitters and seeing where we actually can get a signal in this, which should also give us like some sort of an overall idea what can which part of the nervous system actually do. And I want to show you some data on this a bit later. And then what we also do is like transcriptome searches. So we want to see like which neurotransmitters and neuropeptides do they actually have and how do they differ from like, first of all, the other two species, but then also like the females of different stages. So it could be that an adult dwarf male has like a different neuropeptide complement than the females at different stages. So then just like showing you some of the TEM data that we have. So there was, we started like tracing them, and also like from the literature we could get there is like some sensory cells that are of interest. And the question is always like, how does this animal, so the male has just 68 cells or 68 neurons. How can this animal actually kind of find a female, orient to the female and start like copulation? And one thing we are now like quite, well, not quite convinced, but we at least think would be like a smart idea, would having like this cell, which is highlighted here on the left side, which is the sensory neuron number 15, which is on the dorsal side, and which has like an internal cilium and a microvillar collar around it. So it's quite clear that it's a chemosensory organ as well as a mechanosensory organ, or like a mechanosensory cell. And the idea is now that this cell could, once it's activated, initiate the entire copulation of this animal. And we have reason to believe this because in the upper video, like the upstream, so that means that our nerve, which is shown in yellow, gets like information from a different sensory cell, which is like the pink cell, and which is the sensory neuron number 14. So they are kind of cross-talking quite a lot. And if we go like downstream, then we can actually see that our nerve, like the sensory neuron 15, is talking to the sensory neuron 14, like kind of back-checking what's going on. But also this light green part here is like a muscle in the penis sheath. So most likely it can actually initiate either contraction or arrest of contraction. And this, all this petrol and cyan colored things are different gland cells. And like innervation of gland cells is a super interesting topic because it's not really clear how a nervous system can steer gland excretion. And here we would have like 2 or 3 different types of gland cells, which have like different functions during copulation. So there is also like this interest in, okay, which one can it actually steer into which way? So sorry for that. Here's like some of the immunoreactivity patterns I told you about. So it's basically we have antibodies against certain neurotransmitters, which we put on the animals, and then we check where we can actually get a signal. And the interesting part is with most of them, we got this signal. I mean, all this ring-shaped things, which I hope you can actually see on the screen now, it's all in the circumpenial fiber mass. So this is like this ring that you can also see in the schematic drawing in the middle, which is going around the copulatory organ, and which would also be like the smartest way of distributing the neuropeptides in all different directions in there. So this is also something where I think like, hey, this is actually also quite a cool finding, because when we compare it with the females don't really have all these neurotransmitters in the ventral nerve cord. So they have all these neurotransmitters in the brain. This is now sounding a bit sexist, but it also would be like kind of interesting to see like how much of an input from the brain does the dwarf male need at all, or is it actually that this copulatory region is like an autonomic region to some extent? And then what is like the next steps like for this thing is we want to kind of compare how does it look with the bigger animals. And here is especially like the bigger males. So when we think we have this super small system in the dwarf male, this must be like the essential elements of regulation that the animal needs. I mean, when you are that small, you can't really afford to have extra cells or extra circuits that you can't really use anymore. when you are like bigger, you maybe have like also different things to think about, or you need to outsource some of these parts. So the idea is like, how can evolution more or less like make a small nervous system a big nervous system again, or then of course the other way around, how can it be like condensed again into one form? And then there's like several side questions, like how is gland excretion regulated like by the nervous system? This is something that's actually super interesting, thinking that so many meiofaunal animals are using glands or different sorts of glands for gliding or for sticking to the ground. And we actually know so little about how this is functioning. I mean, how does the animal know when to stick and when to release? And how does it actually do these things? And this is like kind of mind map of all like the topics or all the terms that are like used in our kind of research, like mechanosensory cells, olfactory cells, the different techniques. So we use like a broad range of electron microscopy, confocal laser scanning microscopy, light microscopy, of course. And boiling down, or actually they are all needed to find like the answer to two big questions. And I'm not sure whether we can actually find an answer. This is just what would be really cool to actually get to answer at some point, which is like, what is really needed in a nervous system? I mean, it's just like, what is like the absolute minimum requirement we actually need to have a living animal or annelid that can actually do well in this life? I mean, what is everything kind of boiling down to? And then another thing is like, how constrained is annelid nervous system evolution? So is there anything that an annelid just can't do by getting smaller, getting bigger? Is there any pathways that they normally would take because it's just facilitated, because the gene regulatory network is already set up or can employ it like this? And these are just some of the big questions which we're not really getting that much closer to anytime soon, but we try hard. And that said, like none of this work could have been possible without like the help or like the collaboration with so many different people in so many different labs. So many of them don't have pictures because I couldn't find any on the internet, or I found somewhere for like, maybe I wouldn't like them online. But it's just amazing how many people actually from different fields can work together towards this one question of understanding meiofaunal annelids, or how many people can be convinced that this actually would be a cool approach or cool to find out at some point. So huge thanks goes to all of them, and of course to you for inviting me, but also for listening to this talk now. So thank you very much. And that's it.